Every expectant mother knows that what she eats impacts her baby. And now research shows that is also true for our cows. Maternal consumption of ReaShure during late gestation had a positive effect on the in utero calf, setting her up for better health and potentially even higher milk production once she joins the milking string. Learn more at balchemanh.com/launch and launch your herd for life.
Hello, everyone, and welcome to the Real Science Lecture Series. My name is Scott Sorrell, Director of Global Marketing at Balchem. Today, we welcome Dr. Leeman Kung from the University of Delaware to discuss the potential negative effects on intake and production when it comes to aerobic stability of silages. Dr. Kung is a native of Honolulu, where he obtained his bachelor's and master's degrees in animal science. He completed his PhD at Michigan State in dairy science. After working on bovine somatotropin at Monsanto for several years, he joined the staff in the Department of Animal and Food Sciences at the University of Delaware. Leeman recently retired as the S. Halcock DuPont Professor of Animal Sciences, an endowed professorship where he had a research, teaching, and extension appointment. He also led the department as chairperson for 5 years. His research was centered on ways to improve the productive efficiency of lactating dairy cows through a better understanding of fermentation processes that occur in silage and in the rumen of dairy cows. His silage program has been recognized in the U.S. and internationally, and he is a sought-after speaker at dairy science meetings throughout the world. Dr. Kung, the floor is all yours.
Good morning, everybody. Thanks for being with us today. And thank you, Scott, for the introduction. And thank you, Balchem, for inviting me to speak to all of you today. So as Scott said, I'm going to talk a little bit about wild yeasts and their effects or potential effects on the aerobic stability of silages and total mixed rations. The topics for today are going to include a little bit of a background on the metabolism of wild yeasts and silages. We'll talk about their undesirable characteristics. We'll also talk about specifically how they affect the aerobic stability of silages and subsequent total mixed rations. And we'll talk about some potential direct negative effects on ruminants, including production, intake, and maybe some rumen fermentation issues that they may impose. And then finally, we'll talk about methods to minimize wild yeast in silages and total mixed rations. Okay, so as we start off, I want to let you know that there are a wide diversity of yeasts and silages, and we've known this for a long time. This slide shows the types of wild yeast that were identified in corn silage samples on the left and high moisture corn samples on the right, and from different farms throughout the US, New York, Idaho, Ohio, Washington, West Virginia. And you could see that there's a whole slew of different types of wild yeasts. One of the things that we're aware of, that we're not aware of, is why there's such a diversity of yeasts that are present that differ between types of silages. So as you can see, the types of yeast in corn silage versus the ones in high moisture corn are different. And then we don't understand why the diversity is there even among farms. So they're very, very, as you can see, we have a very high concentration of Pichia anomalo here in high moisture corn, not as much in corn silage. We don't know why that is. But even when you look at corn silage itself, you can see that by farm, there are very few farms that actually look similar with each other. So why that diversity is there is really unknown. We do know that metabolism of wild yeast occurs in two different categories. First of all, we simplistically have divided yeast into what we call fermenting yeasts. And these organisms under anaerobic conditions will convert sugars to ethanol, carbon dioxide, and water. The final result of this is a really inefficient use of glucose fermentation, as it yields only about a 51% recovery of dry matter in a pure culture. Now, the importance of this is that if you are feeding cows, cows not only eat energy, but they eat dry matter. So even though anaerobic fermentation of glucose to ethanol by yeast, even though that process yields a high recovery of energy, it yields a very low recovery of dry matter. And that's not acceptable because, of course, cows do eat dry matter. Now, the other broad category of yeast that we divide things into are yeasts that are called lactate assimilating yeast. So these organisms are yeast that under aerobic conditions will oxidize, not ferment, but they will oxidize and utilize lactic acid to make carbon dioxide and water. And unfortunately for the animal, CO2 and water really doesn't do anything. And the net result is a loss of dry matter. And we know that it is the lactate assimilating yeast, not the fermenting yeast, that are the primary initiators of aerobic spoilage in silages. But again, both fermenting and lactate assimilating yeasts are really undesirable in the lactating dairy world. Now, many of you have seen this slide before, as I've used this in almost all my talks, but it's a good background to set the stage of what fermentation looks like under both ideal and fermentation conditions, followed by good storage conditions. So first of all, we see sugars that are present within the silo. They're released because of chopping. Air is removed from the system because of packing, and sugars over time within the silo will decline. They're declining because the good lactic acid bacteria are consuming those sugars and making lactic acid as an end product. Because lactic acid is such a strong acid, it'll decrease the pH. And also there's a little bit of acetic acid that's made during the fermentation process. The key take home from what you've seen so far in this PowerPoint slide is the fact that there's a lot of things that are happening very early in the fermentation stage. As you can see here, things are moving and changing. And the faster that we can get these things to stabilize and reach what we call a steady state, the more dry matter and energy we will preserve over time. The longer it takes to hit a steady state, the more energy and dry matter we'll lose over time. Now, that's what happens in an ideal fermentation phase. Next, if we keep air out of the silo during storage and feed out, and we will end up with a stable and high quality silage, and we'll have something that looks like this, where basically nothing happens. And as you know, silage can be kept for decades. I've literally seen silage samples at the bottom of cement stave silos that never really got to the bottom. I saw a sample several years ago that was almost 25 years old, and that silage looked beautiful because there was no air that was allowed to penetrate into the mass. There is some heating that occurs during the siling process, but it usually dissipates over time, and we end up with that steady state. Now, this is what happens under ideal and good storage conditions, but we could have this explode on us and cause a problem. And I'm going to show you again the fermentation phase that I just showed you before. But now during storage and feed out, if there is exposure to air, we end up with this happening. Lactic acid disappears, sugars disappear, acetic acid disappears, and the pH goes up. And then, of course, we get a secondary heating event that's occurring during storage or feed out. And of course, this big loss of heating that's occurring here is certainly an indicator that we're losing a lot of dry matter energy, because heat is energy and heat is our dry matter loss. Now, the question that becomes apparent is why is this happening? Well, it's the result of what I call the domino effect, or the chain reaction effect of air on wild yeast in silages. What we see is silage is exposed to air, yeast wake up, and it's the lactate assimilating yeast that wake up that degrade lactic acid. The numbers of yeast increase over time. Highly degradable nutrients are destroyed. Heat is produced. pH increases because the lactic acid disappears. Then a secondary wave of microorganism further spoils the silage. And these organisms are molds and aerobic bacteria. They kind of wake up because the low pH that was keeping them asleep has now disappeared. And so now they're all awake and metabolizing and oxidizing feedstuffs. They cause more heating, and then you end up with massive storage. So if you look at the chain reaction, if you want to ask yourself, well, what can I do to prevent the domino effect or the chain reaction from occurring? Obviously, is you need to prevent the falling of the first block or the first domino, which is exposing silage to air. Now, unfortunately, that is an impossible thing to do in the real world all the time. That is, how can we always keep silage away from air? On a practical basis, that's probably impossible all the time. So then we have to stop the second block or domino from falling, and that is to prevent these yeasts from waking up. And the easiest thing to do there is to decrease their numbers so that there are fewer yeasts within our silages. And with fewer yeast, we'll have less rapid spoilage within our system. This is a graphic pictorial depiction of what happens when silages spoil over time. You can see the increase in numbers of yeasts, temperature increases as well, and the pH increases. And we also usually get a secondary heating peak. And again, this is because of molds and aerobic bacteria. One of the things that I want to point out is the fact that it is often a misconception on the farm that spoiling silages are because of molds, because that's what often people see. It's very difficult to see the yeasts in spoiling silages, but people do see molds because molds have different colors. They're red, they're white, they're orange, they're sometimes yellow. But I will tell you that molds are not the initiators of aerobic instability in silages. They are opportunistic organisms that certainly occur and aid in the secondary spoilage of silages, but they don't initiate the spoilage. Now, this is all important because when you look at the dry matter losses from a good and poor management, and I've got categories here of where major losses of dry matter might come from, which would be respiration, fermentation, seepage, and aerobic instability. If you just draw your attention to the red area here, you can see that even under good management, aerobic instability is responsible for probably 50 to 60% of the total dry matter loss, even within a good silo. And of course, in a silo that is poorly managed, dry matter losses from aerobic instability during storage and feeding can be even more than 60%. So this is an important area that we should be really focusing on to improve farm efficiency, and that is to keep our silages from spoiling aerobically and keeping our TMRs from also spoiling aerobically. Now, of course, all of this comes down to losses, and it's losses of dry matter because of the oxidation of nutrients, but it's also a loss of production because animals eat less, because there's a reduction in intake that's usually associated when spoiled silages or TMRs are fed to lactating dairy cows. And of course, there's the issue of adding increased cost of a ration. Oftentimes, if you have spoiled silage coming out of a silo, the nutritive value might be poor. And so you're increasing your concentrate, maybe your protein, maybe your energy, and that's going to obviously affect your bottom line. And then, of course, there's the potential for other challenges and costs because of potential from mycotoxins, rumen acidosis type conditions, etc. So there's a lot of negative effects of having spoiled silages and spoiled TMRs available for your cows. Now, yeast grow really quickly, and this is an issue for us because it's all about numbers when you talk about these lactate assimilating yeasts. And yeasts have a doubling time of about 1 to 2 hours. And what I'm showing here is the growth of, let's say, yeast in a pure culture. If we start out with 100 yeast at time 0 and measure it over time going through 12 to 14 hours, we can see that very quickly within a 14-hour period, we've gone from 100 yeast to 1.6 million yeast per gram. So replication is staggering here. And it's all a numbers game. I kind of mentioned that in the previous slide, but let's just look at this graph here and say that this is where spoilage occurs. If I start with a really high population of yeast in my silage and I expose it to air, it's going to spoil over time. And it's going to, let's just pick a number, any number, it really doesn't matter. But relatively speaking, let's say 12. So it took 12 hours in this example with high yeast for this silage to spoil. But if we had started with a low population of yeast in our sample, it's eventually going to spoil as well. But hopefully it'll take a longer period of time. And in this example, it took 60 hours to spoil. So aerobic stability of 60 hours for a corn silage sample is about normal of what we would expect in a really good silo. But oftentimes aerobic stability can only be 12 hours in a silo if it's challenged because of different kind of management issues that we're faced with on the farm. Okay, so if we know that yeast are a problem, and we know that it's the lactate assimilators, we also know that it's a numbers game. What kind of numbers do we see out in the field? This is some data that I borrowed from Cumberland Valley, looking at over 700 samples. This is a relatively old slide, so there's probably some new slides out there. But of the 700 samples that the lab had analyzed here, you can see that there's a significant amount of samples that are less than 100,000 colony forming units of yeast per gram, and that would be good. But look at all these samples that are high. And so we've got some samples that have over 250 million yeast per gram, where, as I said earlier, 100,000 is about normal. And so, of course, these silages that are over probably 1 million yeast per gram, they have no aerobic stability. They have all spoiled already. And so it's a very large percent of the whole population of the samples that were looked at. And on a simplistic basis, we know that there is a negative relationship between the total number of yeasts and aerobic stability. This is from a data set from one of the studies we did several years ago from our lab. But you can see that once we reached about 1 million colony forming units of yeast per gram of corn silage, aerobic stability was 0. So that means that this was very unstable material, probably spoiling already. But as we lower the numbers of yeast naturally found in corn silage, we can see the hours of aerobic stability increase. Now, is this a real problem on farms? And I always tell this story to a lot of people that many years ago, I was talking with a company about looking at evaluating total mixed ration saver type products, because we knew that TMRs in the north, across the U.S. and across the world, obviously spoil, and they, especially during warm weather, they spoil. And I remember the rep I was talking to said, "Oh, TMR is spoiling. That's not an issue." And so back in 2005, which is a long time ago, I had a undergrad, Michelle Morgus, go out and sample TMRs in the Northeast region over 2 years. In the first year, we hit 12 farms. In the second year, we hit 18 farms. And we measured how stable these TMRs are. And you can see that in year 1, 8 of 12 farms had stabilities that were less than 12 hours. And in year 2, 8 of 18 farms had stabilities of less than 18 hours. So it's a significant proportion of total mixed rations during the summertime that are spoiling before a 24-hour period. And in the Northeast, at least in our area, in the Mid-Atlantic area of Maryland, Pennsylvania, Delaware, there are not that many farms that are feeding twice a day. By far, the majority are feeding once a day. And so anything that's spoiling in less than 24 hours. So here now we have 12 farms, or all the farms are spoiling within 24 hours in year one. And about 60 or 70% still are spoiling in year two. This could be a potential problem for our animals. Now, why should you be concerned with feeding silages that look like this, that are obviously aerobically spoiled? It's because there are potential negative effects of these yeasts on both silages and total mix rations. We have heating that occurs. Heating can reduce intakes. We have reports in the field from acidosis-like conditions, and also reports of lowered milk production and milk fat depressions and other health problems as well, as you suspect. Here's some data out of Germany. There is a really big negative correlation. The R-squared was minus 0.85, highly statistical significant. And in their data set, they showed that, in fact, dry matter intake in these goats that were fed spoiled corn silage was increased dramatically as the dry matter intake decreased dramatically as the change in temperature of the silage mass occurred. So the hotter this silage got, the more and the less and less intake these goats were eating because of some kind of sensory perception there. We also did a study several years ago where we looked at the effect of feeding a spoiling TMR to heifers. The treatments were either heifers fed a fresh TMR or those fed a spoiling TMR. And the spoiling TMR was basically the fresh TMR that was placed in bins with holes, heated in a room for about 2 days prior to feeding. And when this material was fed to the set of heifers that were fed the spoiled TMR, the temperature of the TMR coming out of those bins ranged from about 90 to 130 degrees Fahrenheit, which is within the range of temperatures that we've actually measured on farm in feed bunks during the summertime. If you look at the corn silage that was made, that was used in this study, you can see that. This slide is out of place here. Sorry. If you look at the fresh TMR, versus the spoiling TMR, this is what we saw. And when you look at the p-values, there's nothing that's significantly different. So the dry matter is the same, the protein's a little low, but these are heifers, remember. ADF is not different, NDF is not different, NDFD is not different. Starch the percent is really not different, and starch D is not different. So on a technical basis, when you're looking at, is the spoiling TMR, can we pick up differences in nutrient value? The answer is really no, but we can pick up differences is in the fermentation profiles of the fresh versus the spoiling TMR. So we did see a higher pH in the spoiling TMR. We saw less water-soluble carbohydrates. We saw less lactic acid. We saw less acetic. We saw more ethanol. But the big thing that we saw was more yeast. And these are log values here, 5 versus 7.2. On a numbers basis, log 5 is about 100,000 yeast per gram. Log 7.8 is about 66 million yeast per gram. So the really big difference was in the numbers of yeast. And we know from previous in vitro studies that yeast don't die in rumen fluid, and they actually maintain themselves. These heifers were rumen fistulated, so we were able to take samples from them. And exactly what we saw was that we could pick up spoilage yeast at a level that was almost equivalent to what was in the total mix rations in the spoiled TMR versus the fresh TMR. But now these are yeasts in the rumen of the heifers, not just yeast in the total mix ration itself. And because of that, what we see is about a 12 to 13% drop in dry matter intake. So this is different from the fresh TMR, about 10.5 kilos per day versus almost about 11.6 kilos per day. So the question that pops up then is, what causes the negative effect of feeding spoiled silages and TMRs? What's the mechanism of this? Could it be production of toxic end products from, for example, yeast do present and produce mycosins? Are there alterations in nutrient value? Could it be changes in fatty acid content of the yeast cell walls? Could there be changes in biohydrogenation of fatty acids? Or could there be organoleptic effects like taste, smell, or just the hot feel? Simplistically, do these animals smell and taste this, and does it smell bad or taste bad to them? We really don't know the specific reason why animals are dropping in intake when they're eating these spoiled feeds. But let me show you, just from a perspective standpoint, the numbers of yeast that we're potentially feeding, and how that relates to, for example, an additive. So what I have here is I made a little table of levels of yeast that are in the diet. And so let's just say I have a sterile yeast that has no yeast in it, so it's 0. The 1x is log 5. So remember, I said initially that the average number of yeast in corn silage is about log 5 or 100,000. So that's this, okay? I then did what happens if I'm at 100x to log 7. And we saw log 7 yeast in the previous study, right? In the TMR, that was like log 7.3 or 8 or something like that. And what happens if we go up to a log 9, which would be the some of the samples that you saw in the CVAS slide that were way up in the high level of the 200 million yeast per gram. So we've got a wide range of yeasts and silage presented here. And now if you're an average cow and you're eating about 66 pounds of 35% dry matter corn silage, so I just took that number out of the air, and you put it into the average capacity of a 1,500 pound cow, you can do the calculation that these are the concentrations of yeast that you would find within the rumen of a lactating dairy cow. Okay, log 4, log 6, log 8. And then what I did was I made the theoretical calculation of how much good yeast from a yeast additive. Okay, so we're talking now something like Levucel, where you have a live yeast count that you're feeding good yeast for beneficial effects to the animal. How much Levucel or good yeast would you have to feed to attain the numbers that we're looking at here? So if you're at normal corn silage of log 5, the yeast that's bringing to the table to the cow is the equivalent of about 0.15 gram of good yeast per day. On the extreme, if you had corn silage that had log 9, or these 200 million yeast per gram, you're bringing the equivalent of 1.5 kilos or 1,500 grams of bad bacteria to that animal. So, simplistically, if you can imagine that a very small amount of good yeast can be beneficial to your cow, can you imagine what a really high concentration of potentially bad yeast could have to your animal? And of course, you can let your imagination go wild there. One of the studies that we had done several years ago as well was Mateus Santos, which was a graduate student from mine out of Brazil, actually isolated a wild yeast out of high moisture corn. He grew that wild yeast up in pure culture and added it back to have different log concentrations that were 4.4, 6.4, 8.4. And you can see these log concentrations again are numbers that I showed on the previous slide, right? And that we can find in TMRs that were fed to our heifers. So these numbers are not out of the ordinary. There are real numbers that you could find out in the field. And he put back this isolated lactate assimilating yeast into in vitro cultures and looked at fiber digestibility. And one of the things that he saw was that the more wild yeast that he added to his in vitro culture, the lower was the NDF digestibility. Now, does this occur in the rumen of a live cow? We don't know. We only did this in vitro, and no one has ever repeated this. But there is a potential then that what we can see here for having a direct effect on metabolism within the rumen. If wild yeast, especially the lactate assimilators, are an issue for our cow, one of the questions that then pops up is, how do we minimize wild yeast in silages? Well, it's the back to basics, back to management thing, right? We need to asile our silages at optimum dry matter, have correct particle size, fill fast, pack quickly, use good plastic and weights, use defacers, use covering pads, use gravel bags to keep the air out, all the things that we talk about for good management. That's how we keep wild yeast and lactate assimilators, because it's the air penetrating the mass that allows them to really grow. Obviously, the other thing that we can do is to use additives that are designed to minimize wild yeast, and those would be, Buchneri based inoculants and organic acids as well. And of course, excellent silage management is needed to maintain this high quality silage for sure, right? From the silage standpoint, we know that the family of L. buchneri microorganisms that contains not only Lactobacillus buchneri, but also contains organisms like Lactobacillus dialavorans, Lactobacillus hilgardii. These microorganisms are heterolactic bugs that basically take a little bit of lactate anaerobically and can convert it to moderate amounts of acetic acid. And we know that acetic is very high in antifungal properties. The other thing that we could do is that we could add organic acids at ensiling, or we can use organic acids as TMR savers. So having said this, how do these acids kill or inhibit these lactate assimilating yeasts? Well, that's a really good question. This is all based on the fact that a little bit of chemistry, and it's not too hairy, but let me get you through this, is that all acids have something that is called the pKa. And the pKa of an acid is equal to, when pKa equals pH, which would be right here, we would have 50% of that acid would be in what we call an undissociated active form. And if the pKa is equal to the pH, 50% then is in the inactive or ionized form, all right? So the thing that's important to have killing or moderating activity of acids added to silages or TMRs is that you want to maximize the amount of this acid that is in the active undissociated form. But as the pH moves higher above the pKa, we end up with more and more ionized, dissociated, inactive form of the acid. So I can tell you that, just take an example. If we had an organic acid like acetic acid, we put it into a silage that had a pH of 7, I will tell you that all of the acetic acid in that silage would be inactive at a pH of 7. Okay. Now, if you put it into a silage that had a pH of 3, probably almost all of it would be in the active undissociated form. How do these things actually work? Let me show you. First of all, what I have is a picture of a yeast cell wall. This is its membrane. This is the internal side of the yeast. This is the outside of the yeast. The importance of having undissociated organic acids to kill yeast is this. It is only the undissociated form that can penetrate the yeast cell wall and get into the cell itself. These types of, if it was dissociated, it would not be able to get in. What happens then is that as the dissociated form enters the cell wall, it dissociates into an anion and hydrogen. The first thing that happens is that anions are toxic to the cell. So as it accumulates, it can kill the microorganism. The second thing that occurs is that the excess hydrogen ion now lowers the internal cell pH of the microorganism and affects enzymes. So it takes it out of its comfort zone. The third thing that happens is that as these hydrogen ions accumulate within the cell senses this and it wants to get rid of that hydrogen ion because it's affecting its enzymes. But in order to get the hydrogen ion out of the cell, it uses energy, ATP, and then it puts itself into a fetal cycle where all the energy of the cell is being forwarded to getting rid of the hydrogen instead of using that energy to grow and make proteins and enzymes and to do metabolic functions that are important for its survival. And so the cell gets rid of ATP. The other thing that organic acids do is that they alter the disruption of organic acids through the cell, nutrients, amino acids, minerals, etc. So these organic acids have multiple ways of inhibiting a lactate assimilating or wild yeast. For Buchneri, I've shown this in many different presentations before in the past as well. Buchneri takes a little bit of lactic acid. It'll take acetic acid and improve aerobic stability. Now, one of the things that we did recently was trying to look at how do we improve the aerobic stability of TMRs and not just silages, because we know that people don't just feed silages anymore, right? We feed silages mixed into total mix rations. So we had a control and additive treated corn silage from farm 1, and we looked at the corn silage stability. So stability of the control corn silage in this farm was 50 hours. The additive treated corn silage was 200 hours. So this is just the corn silage by itself, all right? And that's different. Then what we did was we used ingredients from 4 different farms, but we used the corn silage from only farm 1, and we made total mix rations at all 4 of the farms. One of the interesting things that we saw was we did see a carryover in farm 1 where the total mix ration was that had the treated corn silage was still significantly better than the control. But for the other 4 farms, there was really no effect on the stability of the total mix ration. And so that's a puzzling result that we saw there. The question then becomes, why isn't there always this carryover effect of additives into the TMR from the silage? And the answer is actually quite simple. That is that there are TMR components that add buffering capacity and raise pH. It's actually quite common for farms in our area of the region to have total mix rations where the pH is about 4.8 to 5, remember? And remember that we said that as the pH goes above the pKa, we get more and more inactive forms of the active compound. So we know that in a total mix ration, you're adding halage that has a really high pH. You're adding probably sodium bicarbonate. So it's not surprising that the total mix rations have a pH of 4.8 to 5, which are much higher than corn silage by itself, which would be something like about 3.8 in our region. Another issue that we see quite commonly is that TMR components may also bring wild yeast. Not only can it be coming in from spoiling silages and wet brewers grains, but here's an example of haylage that's got a lot of mold and yeast in it, distiller's grain, brewer's grains. All of these can actually bring in spoilage microorganisms. So what this slide shows is the aerobic stability curves of total mixed rations made with either no spoiled silage, which is the yellow or orange line that you see here, and then with the curves of heating. And you can see that adding even only 10% of spoiled silage to this total mixed ration completely destabilized it, and it spoiled within a 24-hour period. So one of the important things that I always remind people is that, the leftover feed in the feed bunk probably should not be mixed into the new day's feed, because it could destabilize everything, not only the old, but the new total mixed ration as well. So in summary, we know that all yeasts are undesirable in silages and total mixed rations. Lactate assimilating yeasts are the primary initiators of this spoilage. We do know that feeding spoiled silage can have detrimental effects on the animals. And we know that practicing good silage management, for instance, packing and density and plastics, etc., and additives are all important to try to help minimize the numbers of these undesirable organisms. And of course, maintaining high quality silages and total mixed rations, especially in warm weather, is not just as simple as using silage additives, but feeding management and sources of yeast contamination should also be considered. And with that, I thank you.
All right. Thank you, Lehman. Before we get started answering questions, we'd like to share a brief video. Then we'll be right back to answer the questions submitted during today's presentation.
The heat of summer is coming, and it can have a big impact on your cows. NiaSure Precision Release Niacin is the perfect complement to traditional heat abatement strategies to help keep her cool from the inside. Using Balchem's proprietary encapsulation process, NiaSure delivers 8 times more bioavailability than raw niacin, leading to an increase in sweating and vasodilation to reduce internal body temperature and support maximum productivity. Learn more at balchemanh.com/cool and keep her cool from the inside.
All right. As a reminder, you can still submit questions through the Q&A tab at the top of your screen. Lehman, I know you said early on that there are a lot of different strains of wild yeast out there. Are some more problematic than others? I remember about 15 years ago, we started hearing a lot more about issues with wild yeast. I think that was in the Northeast. What can you tell us about that?
Yeah. As far as we know, as I said in the talk, the lactate assimilators are the ones that are really the initiators of aerobic spoilage per se. We don't have any indication or idea really of any specific yeast itself that might have a negative direct effect on an animal, so we don't have that information. The research on wild yeast is very, very limited. It just hasn't been a big funding area for people. And so it's a tough call to say, that any one specific organism is the culprit. And so I kind of tend to lump them all together. And as I said in the beginning of the presentation, the take home for me is that all types of yeast are bad, right? Because they can be bad from a fermentation standpoint, but they can also be bad from a aerobic stability standpoint as well.
Very, very well. Dr. Hutchins is asking, if I were to use an organic acid source in a TMR, what level of acetate or propionate is recommended?
Yeah, that's a really hard one to say, again, because it depends on what the pH of your final TMR is, right? And as I said, shown in the data, if for whatever reason your total mix ration that you're putting your silage and your additive into, if that pH is on the high side, then the amount of active ingredients you need has to be very, very high, because otherwise it's all in an inactive form. So if you're the one thing I would always look at is to try to determine what is the pH of your whole total mix ration, and then if it's kind of in that 4.5 range, you probably would need I would say about 3% total acetic acid to 3.5% acetic acid in your corn silage to have good stability. But again, if that corn silage is going into a total mix ration that had a pH of 6.6 for some reason, even that might be not enough to stabilize the whole TMR.
Okay. Dr. Jim is asking, what causes some silage to have a high acetate acid content, not treated with butaneri? Lactic and acetic could be close to a 1-to-1 ratio. In any negative effect on cows?
Yeah, so we know that silages, especially corn silages, and actually alfalfa silages too, when they're put up really wet, there are microorganisms that really go to town, especially heterolactic acid bacteria that can produce acetic acid, but also enterobacteria. So enterobacteria is another class of microorganisms that really like wet conditions. And so corn silage at 28-30% dry matter, they really go to town, and they can fuel the really high concentrations of acetic, even though that silage may not have been treated with butaneri. And so one of the things as a recommendation is obviously which has always been, is to try to keep these silages away from being put up too wet because of that potential enterobacteria problem. And of course, with haylage, it's another problem because of Clostridia. But certainly high wild acetic acids could cause intake depressions. And we see that in the field, right? It's the number one issue I see with people feeding wet corn silages is usually intake depression, and it's probably associated with high acetic.
All right, very well. Fernando wishes, he liked the presentation, wishes he could see it again. Fernando, you can. We will place it in 48 hours on balchem.com/real science. So go there in 48 hours, you'll be able to watch this again. Ricardo is asking, will the live yeast fed in an additive show in the yeast count analysis of a feed lab? And if so, what is the easiest way to interpret those analyses?
Yeah. Live yeast added can be found, can be picked up in a lab analysis, but the amount of live yeast that you're adding from an additive is probably not enough that it would be above the background level of what you had in a normal total mix ration. So you wouldn't be able to use a yeast count in a TMR or in a feed to predict or to verify that you added the right amount of yeast. You can't do that.
All right. Sayer is asking, how much does the temperature affect the doubling time of yeast? Do you have any figures?
Yeah, not off the top of my head, but certainly that's a very good question, because as temperatures start dropping below, 60 degrees or so, obviously these yeasts their metabolism will start to shut off. And of course, this is why we have less aerobic heating problems in the wintertime, right? Because even though the yeast are there, they're not doubling anywhere near as quickly. But, so I don't have that number off the top of my head. I can find it, and maybe we can add it someplace in a question and an answer.
All right. Maxim, when we have warm weather and add the good yeast culture to the TMR, is it provocateur to heating TMR or not? and we have the same bad effects from good yeast or not?
No, yeah. So
Okay.
that's a good question. But when we add these good yeasts are not lactate assimilators. So adding good yeast will not cause your TMR or your silage to spoil more quickly.
Okay, next question comes in from Brian. You mentioned earlier that there is a wide diversity of yeasts and silages that were variable by feed and by farm. Do you see when a farm has a high level of certain strains of yeast continue to see the same strains year after year, or is it each year a new set of circumstances?
Good,
Interesting.
good question, and we do not have that data.
All right.
Yeah. I will just point out too that a common question that comes up is, do high yeast counts at the time of harvest have an effect on subsequent aerobic stability? And the answer to that question is we probably don't know for sure, but I'm going to go out on the limb and say that yeast counts themselves at the time of harvest probably are not a good predictive number of what's going to happen later. It's more of what happens during silo management that can really affect aerobic stability at the end. Because you could have relatively low numbers of yeast on the standing crop, but if you had that followed by poor management, slow filling, poor packing density, et cetera, that'll just blow those numbers up and cause very bad aerobic stability. So I don't think that yeast numbers at harvest are predictive of stability during feed out.
Okay, very well. Mark is asking, would bacillus at a high dose in a silage additive inhibit wild yeasts?
Don't have the answer to that question. Really don't know. The only thing that you would hope for is to have a bacillus strain that maybe produced some type of antifungal compound to control the lactate assimilators. But I don't know of any such product that exists.
All right. Mac is asking, acetic acid and propionic acid have pKa's that are somewhat close to each other. Why does acetic work better on yeast than propionic?
That's a good question. If you take a look at the slide where I showed how organic acids actually affect the internal mechanism of a cell, from a simplistic standpoint, my answer is that acetic probably is working a little better than prop because it's probably having the combination of factors affecting the yeast more strongly than prop does. And I know that maybe that's not the best answer, but we don't know specifically why different organic acids, even though they all may have pretty good killing mechanisms and have pretty similar pKa's to each other, we don't know how they're specifically different from each other. It's not just pKa and pH. There are other factors that can occur that can affect their efficacy as well.
All right. Jim wants to know, he says, years ago at the Wisconsin Marshfield Experiment Station, Dr. Howard Larson developed an interpretive scale for mold counts on silages and TMRs. Has such a scale been developed for lactic assimilating yeast? And what is the current state of the question with regard to interpreting fungal counts on silages?
Yeah, no, so that has not been done with yeast. There is no scale. The only kind of thumb rule that we have had for many years is that as an indicator of what's going on is that normal corn silage should have probably no more than 500,000 CFU per gram, and that once you get past 1 million CFU per gram, that means that you potentially are going to have problems. But other than that, there is no better yardstick for us to go by.
All right. Jim's also asking, why is any given alfalfa silage much more stable than any given corn silage?
Couple of reasons, but that's a really good question. A couple of reasons. One, first of all, when you look at corn silage and its buffering capacity, right? It doesn't have the buffering capacity that alfalfa has, and alfalfa also, because of that buffering capacity, usually ends up making a little bit more acetic acid than corn silage. And so the stability of that alfalfa usually is a little bit better than corn silage, because usually the acetics are a little high. Usually the total acids are a little higher in alfalfa because it is buffered and it's going through a prolonged fermentation, trying to stabilize the system, but because the pH is high, it's taking a longer period of time to do that.
Okay. Caroline is asking, why did we get higher propionic acid while ensiling than lactic acid?
We don't usually get higher propionic
Usually.
acid.
Yeah. And if that were to happen, maybe that's what they're asking. What would have caused that?
Or what would have caused a silo to have high propionic acid?
Yes, that's
I
my
have only
interpretation.
seen high propionic acid in silages that have gone clostridial, because of Clostridia. And usually, propionic acid in silages is usually not detectable at all. If you do detect it, maybe you will find 0.1, 0.2% on a dry matter basis. In clostridial silages, sometimes you see it 0.5, 1%, 1.2%. But I've never seen I've never seen any silage with a propionic acid higher than probably 1.2%.
Okay. All right. Jean-Philippe is asking, should we always use L-butyrini in silages, or we should only use it when there is a risk factors associated with heating?
Yeah, I think L-butyrini and its family of microorganisms, all the Hilgardii, dialavorans, combination type products. I think the latter is true, that you should use them if you have a potential issue that's coming up. I don't think it should be used all the time. For example, I would not use a buchneri type product in silages that we know is going to go up wet. So anything less than 30% dry matter, I would probably not put on L-buchneri type product, because it's going to naturally go through an acetic type of fermentation that's going to stabilize it. And the other thing that I have seen in many silages, experimentally anyway, is that if you add L-buchneri type products to really wet silages, you will more than likely end up with a lactate to acetate inversion, where you actually will produce more acetate than lactate. And potentially at the end of the day, that could be problematic for the cow.
Okay. Matthias is asking, how can you interpret differences between active and inactive yeast during ensiling? And do you have any additional tips besides silage temperature?
Not sure I understand that question.
Yeah.
Inactive or active yeast? I mean, I'm not sure I understand that question. Sorry.
Yep. All right. Ricardo's asking, what is the best way to handle samples that go to a commercial lab when trying to study wild yeast counts in silages?
Yeah, that is a problematic issue that we have, is that because yeast multiply very quickly in warm weather, there's always the chance that when you take a sample at the farm, that by the time it actually gets to the forage lab, you always are wondering, is it representative of what I took at the farm? So in the perfect world, samples taken at a farm should be taken quickly, put on ice someplace, air compressed out of the bag, shipped on ice, not frozen. Do not freeze the sample, but ship with ice packs to keep it cool, and get it to the lab as quickly as possible. So that's in the perfect world. And it's really hard to do that all the time if you have a lot of samples. I know it's going to cost more money because you're shipping with ice packs and you're shipping overnight mail. Everything increases the cost, but if you really want a representative number, that's what you need to do.
Okay, the next question comes in from Mario. Sounds like he's putting high moisture corn into tower silos at 18 to 23% moisture, getting a bad result in the summertime. What should the minimum moisture content be for harvesting high moisture corn?
Yeah, I mean in general, ground high moisture corn probably should be put up at about 28. 27 to 28. 18, I'm going to tell you at 18% moisture, there is not enough active water moisture for most microorganisms to grow at all. And so you're right at kind of the no man's land at about, for me, about 22 to 23 is right at the bottom line where you still have enough metabolic moisture for some lactic acid bacteria to grow and ferment a little bit, and make a little bit of acetic acid to stabilize that high moisture corn. But basically what's happening is that when you're putting stuff up at that 18 to 22% range, you're not allowing the natural fermentation to occur. And so there's not enough natural acid being produced to inhibit the yeast. And then that's where you're going to end up with a really terrible result of moldy, rotten, high moisture corn. Yeah. And that's especially true if it's going up whole shelled and not ground, because even in whole shelled corn at 23, most of the moisture is not available for microorganisms because the moisture is in the kernel, and because the kernel hasn't been ground, it's not available to the microorganisms that are on the outside of the kernel.
Yeah. All right, Jim is asking, so adding anhydrous ammonia to corn solids to extend the fermentation would be a good idea for increasing stability, right?
If you were in the late 70s, early 80s, the answer might be yes. But for today, the answer is, on a safety and a practical basis, I would never be putting on anhydrous. I mean, I actually did my whole PhD thesis on anhydrous ammonia corn silage. So I know anhydrous very, very well. And anhydrous raises the pH up. It makes a lot of it prolongs the overall fermentation, so it makes more acetic. But not only is there more acetic in anhydrous, the ammonia itself is a really good yeast killer. So this is why anhydrous ammonia treated corn silage was so stable. It had excellent aerobic stability. But in today's environment, can you afford to be messing with it from a safety standpoint and a cost standpoint? And I would say the answer to that is probably no.
Yeah. Dr. I'm not even going to try to say it. Sorry. Are all yeasts harmful?
Probably from a practical standpoint, at the beginning of my lecture, remember I said that all yeast are undesirable because of what they are doing to the end product of our fermentation process, right? So they're either causing a ethanol type of fermentation where we're losing dry matter, which is undesirable, or they're the lactate assimilators that are causing and initiating aerobic stability. So from that standpoint, all yeast are bad. All right. Yeah.
Okay. Got time for one more, and then we'll call it a day. Is there any practical test on farm to analyze yeast count or pH test?
You know what? I'm gonna go back to the last question, and maybe she asked this question because of the good yeast, and I kind of I missed that point there.
Mm-hmm.
But so not all yeasts are bad if they're good yeast, right? So the Saccharomyces cerevisiae that people are adding as beneficial direct fed microbials, Those types of yeast added to a total mixed ration are okay, right? But in fact, if you had added Saccharomyces cerevisiae as a good yeast, but you tried to add it at the time of fermentation, that would actually end up being a bad yeast because it would still ferment, and that would be, unwanted in your system, right? So good yeast added as a post facto additive into a total mixed ration are okay, but yeast coming from the plant are all pretty much not wanted within our system. Sorry, could you?
Yes. So Mario's asking, is there any practical test on farm to analyze yeast count and pH?
pH, yes, you can have little pH pens that you can get from Amazon for $75, so you can measure pH there. There's no quick yeast test on the farm. Everything takes a minimum of 48 hours to actually grow the yeast and identify them in the analytical labs.
Mm-hmm. All right. Very well. Well, thank you, Dr. Kong. This has been very interesting. Thank you, everyone, for attending today's webinar. If you have additional questions, please submit them to anh.marketing@balchem.com. The Real Science Lecture Series of webinars continues on September 12th with the High Fertility Cycle presented by Dr. Paul Fricke from the University of Wisconsin. Visit balchem.com/real science for more details and to register for all future webinars. Balchem's podcast series continues to offer a deeper dive into our webinar topics. Log on to your favorite podcast platform and search for Real Science Exchange or visit balchem.com/podcast. If you want a really cool Real Science Exchange t-shirt, just subscribe to the Real Science Exchange. Send us a screenshot along with your address and shirt size to anh.marketing@balchem.com, and we'll get that off to you right away. And on behalf of Balchem and Dr. Kong, thank you for joining us today.